Hello everyone, welcome back to Anatomy Weekly. From today's class, we are going to learn about the nervous system. The human nervous system is an extremely efficient, compact, fast and reliable computing system, yet it weighs substantially less than most computers and performs at an incredibly greater capacity. The nervous system receives the sensory stimuli from the internal and the external environments, integrate the sensory information, coordinate and control the voluntary and involuntary body activities, assimilate the experiences as memory, which help in learning and developing intelligence. The memory in turn helps us to establish pattern of responses in the future based on prior experience. Last but not the least, the nervous system helps in programming of the basic instincts. The nervous system consists of three basic functional types of neurons: sensory, motor, and interneurons. The sensory neurons detect the stimuli and the motor neurons send commands to the effector organs. The interneurons lies in between and they confer on the nervous system its prodigious capacity to analyze, to integrate, and to store information. The sensory stimuli, which is the efferent impulse, received from inside or outside the body, are correlated within the nervous system, and then the coordinated motor response, that is the efferent impulse, is sent to the effector organs.
Anatomically, the nervous system can be divided into two parts: the central nervous system, which consists of the brain and the spinal cord, and the peripheral nervous system, which includes all the neuronal tissue outside the central nervous system, such as the cranial nerves, the spinal nerves, and ganglia associated with them. Functionally, the nervous system can be divided into two parts: the afferent division and the efferent division. The afferent division brings sensory information to the central nervous system, whereas the efferent division carries motor commands to the muscles and the glands. Now, out of these, the efferent division has somatic and visceral components constituting somatic and autonomic nervous system respectively. The somatic nervous system provides the voluntary control over the skeletal muscle contractions, whereas the autonomic nervous system innervates involuntary structures such as the heart, smooth muscles, and some glands, and thus provides an involuntary regulation of smooth muscles, cardiac muscle, and glandular activity. This highly specialized and complex nervous system consists of two principal categories of cells: the neurons and the neuroglia. Neurons are the basic structural and functional units of the nervous system. They are excitable cells which are specialized for reception of stimulus and the conduction of nerve impulses. Neuroglia, or the glial cells, are supportive cells that support the neurons both structurally and functionally. The neuroglia are 5 times more abundant than the neurons and account for more than half of the weight of the brain. Now, in today's episode, we'll be talking about neurons in details, and in subsequent episodes, we'll be explaining the neuroglial cells.
The neuron has two regions: the cell body, which is also known as the perikaryon or the soma, and the neuronal processes. These neuronal processes, which are also called neurites, are of two types. These are called dendrites and axons. A neuron may possess one or several dendrites, but it has only one axon. The total volume of cytoplasm present in the neurites is much greater than in the cell body proper. The size and morphology of the cell body vary and are very specific to specific regions and functions of the nervous system. The most prominent feature of the soma is the nucleus, which is usually round or oval, centrally placed, and possesses a fine chromatin network and well-defined nucleolus. The cytoplasm of the perikaryon is rich in free ribosomes and rough endoplasmic reticulum. The cisternae of the rough endoplasmic reticulum form numerous close, parallel configurations which, when viewed with light microscopy, stain dark blue and are frequently referred to as Nissel's bodies or Nissel's granules. The Nissel's granules are absent at the part of the cell body from where the axon begins. This area is called the axon hillock. The Golgi complex is usually near the nucleus and is responsible for the modification and packaging of various proteins, enzymes, and chemical messenger molecules or neurotransmitters. Numerous smooth endoplasmic reticulum are also present in the neuronal cytoplasm. Many of these are located just deep to the plasma lemma as hypolemmal cisternae, believed to be responsible for storing calcium. The energy requirement of the neuron is met by the presence of numerous mitochondria which are distributed throughout the cell body. The cell body is also rich in endosomes and lysosomes, as well as various types of vesicles that are being carried to and from the processes. The cell body also contains numerous inclusions or non-living substances such as melanin pigments, lipofuscin, and lipid droplets. The melanin pigments are present in cells only in certain locations, such as the locus ceruleus and the substantia nigra. Lipofuscin is believed to be the indigestible remains of lysosomal activity, and since it is known to increase as a function of age, it is frequently referred to as the age pigment. Cytoskeletal components of neurons consist of microfilaments, neurofilaments, and microtubules. The microfilaments, composed mostly of filamentous actin, are associated mainly with the cell membrane and aid in elongation of axon or dendrites and in the movement of the plasma membrane. Neurofilaments are the intermediate filaments and form long filamentous threads known as neurofibrils. These are less than 2 micrometer in diameter. Neurofilaments assist in the regeneration and development of axons and dendrites. Microtubules are elongated tube-like structures that possesses microtubule-associated proteins such as MAP2 in the cytoplasm and dendrites and MAP3 in the axon. Microtubules provide a scaffold-like structure to maintain the cell shape and aid in the transport of materials to and from the soma.
Dendrites are relatively short, branching extensions of the cell body. Each neuron may have a number of dendrites. Often the surface of some dendrites is increased due to the presence of dendritic spines. The dendrites receive signals from specialized receptors, axons or other neurons, and transmit these signals towards the cell body. Moreover, since dendrites are usually highly branched structures, they can receive information simultaneously from many different sources. On the other hand, the axon is divided into 3 regions: the initial segment, the axon proper, and the axon terminals. The initial segment is the short transition between the axon hillock and the axon proper. A characteristic feature of the initial segment is the thickening of the axolemma. It is believed to be indicative of the presence of voltage-gated sodium channels. The initial segment is the primary region of the axon, responsible for the initiation and propagation of action potentials. Its length is about 30 to 50 micrometer. The axon proper is a long process, and in some instances it can exceed up to 100 centimeter. The diameter of the axon proper remains constant for much of its length. Axons may possess a myelin sheath that begins at the axon proper and stops at the axon terminal. This myelin sheath is present in discontinuous segments. Each myelinated segment is called an internode. The discontinuities of myelin between the adjacent internodes are referred to as nodes of Ranvier. Occasionally, the axon proper has branches which are known as collaterals that diverge from the main axon at right angles. At its terminus, the axon may arborize, forming numerous axon terminals which are also known as boton terminox or end feet. Now these axon terminals permit a single axon to make synaptic contact with numerous other neurons or muscles or gland cells. The exoplasm does not possess ribosomes or rough endoplasmic reticulum, but there is presence of mitochondria, smooth endoplasmic reticulum, vesicles, neurofilaments, and microtubules. The material produced in the cell body is transported by the assistance of microtubules and their microtubule-associated proteins. Such transport from the cell body to the axon terminus is called as anterograde transport. Now some materials may be conveyed in the opposite directions towards the cell body, and then this process is referred as retrograde transport. Collectively, anterograde and retrograde transport are referred to as axonal transport, a process that is dependent on the utilization of the ATP.
Now a synapse is a specialized intercellular junction that permits communication between the neurons in the central nervous system or a neuron and an effector cell, that is another neuron, a muscle cell or a gland cell in the peripheral nervous system. Now we shall talk about the synapse in detail in subsequent classes. Let us talk a little bit about the classification of neurons. The number of processes a neuron possesses permits a classification of these cells into the following categories: unipolar, bipolar, multipolar, and pseudo-unipolar neurons. The unipolar neurons possess no dendrites and have only one axon. These types of neurons are rare in vertebrates. Bipolar neurons, such as the ones in the olfactory mucosa, possesses an axon and a dendrite. An additional type of neurons, the pseudo-unipolar neurons, which are sometimes also referred to as unipolar neurons in the vertebrates, start out during embryogenesis as bipolar neurons. The axon and dendrite of this neuron fuse into a single process that later bifurcates. The neurons of the dorsal root ganglia and the ganglia of the cranial nerves are representatives of this category. Multipolar neurons, such as the motor neurons of the spinal cord, have a single axon and two or more dendrites. So guys, that was all we had for you today about neurons. In the next class, we'll talk in detail about synapses. Stay tuned to Anatomy Weekly. Keep watching, keep learning. We'll see you next time. Thank you.